So let's spend some time looking at the mechanism of going from a diploid cell to a haploid cell to make a gamete. And this is a process called meiosis. It's a type of cell division. All right, very special cell division. In most cases, the vast majority of your cells are not going to do meiosis. Only a very special set of cells called the germline cells, okay, these are the progenitors of your gametes, are going to undergo this process to make sperm or egg. All right. Now, again, the key is that you're going to make a copy of your DNA, just like you did in mitosis, and then you're going to go through 2 cell divisions to distribute that DNA among 4 cells, each having only half the amount of DNA as the original parent cell. Now, we're going to go through this, and it breaks down in stages much like mitosis does. If you understand mitosis well, this is much, much easier to understand. So you may want to go back and review mitosis after you listen to this, and then listen to this again. Now, this is going to be a focus on the mechanisms of meiosis, and then what we'll do is another recording following this that focuses on the kind of the results and analysis comparison of mitosis and meiosis. All right, so what we're going to see in a summary, meiosis, the first cell division, is going to separate homologous chromosomes. All right. Now, the second division then, the chromatids, the 2 sister chromatids that make up each one of those chromosomes, are going to separate. And now you're going to have 4 daughter cells that are haploid. All right. So let's look at this. So here we go. We're starting with a homologous pair of chromosomes in a parent cell. You can tell because of the same length. They're going to duplicate themselves just like we do in S phase of the cell cycle. Now, they're going to separate from each other. So now we haven't pulled any chromatids apart from each other. We've actually lined up homologous chromosomes on the metaphase plate instead of having all the chromosomes line up individually on the metaphase plate, and that's going to be the big difference. So Meiosis One then creates 2 new cells. Each has 1 chromosome in it, and that 1 chromosome then will go through Meiosis Two, and that will look very, very much like mitosis, where the chromosome, the duplicated chromosome, will separate, the 2 chromatids are pulled apart, and now you get 4 cells with 1 chromosome each, where the parent cell had 2. We've halved that number. All right, so Meiosis One is preceded by interphase, so just like in the cell cycle for a mitotically dividing cells. Right? Now, the chromatids are the same. All that is the same. The setup's the same. The key here is going to be in Metaphase 1, where the homologous pairs line up to each other. Now, each division is broken down into prophase, metaphase, anaphase, and telophase, just like mitosis. But the way you distinguish between what you're talking about, whether it's mitosis or meiosis, is the Roman numeral. So prophase 1 is the first cell division of meiosis. Prophase, now you're talking mitosis. Prophase 2, talking about the second cell division of meiosis. So when you're talking about this, if you leave out the Roman numeral, you are talking about mitosis. Don't make that mistake if you intend to talk about meiosis. So meiosis is always going to have a Roman numeral following the stage of the cell division. So Meiosis 1, Prophase 1, Metaphase 1, Anaphase 1, Telophase 1. We can use PMAT again, no problem. And what we're going to see is that we're going to front load some slight differences, and once we have that down, this thing's going to proceed pretty much like mitosis. So in Prophase 1, we're going to see a breakdown of the nuclear envelope, right? We're going to see a special event occurring, which is called crossing over. This is when homologous pairs are going to exchange pieces of their DNA. And this is amazing stuff, which we'll look at a little bit more detail. Spindle forms. It's like set up for cell division, just like mitosis. Okay? So let's go over each of these in turn as we started with Prophase 1. So this is really where we've got most of the action going on. Chromosomes condense. Synapsis forms. So we get literally the homologous chromosomes lining up. And this is the idea, again, that we talked about. Chromosomes have loci, like they have locations for genes. And because those genes are on the same location, independent of what chromosome it's on is from your mom or your dad, like when they line up and then exchange pieces, what we call these chiasmata. Let's look at this picture again. So you can see these chiasmata, these literally crossing over events. I'm going to break the blue version of this chromosome, and I'm going to break the red version of this chromosome, I'm going to exchange those pieces. As long as I break them in the exact same space, they cross over, and now I'm going to have a chromosome that is partially red and partially blue. So if you understand this idea of ploidy and a diploid organism well, what we're saying there is the chromosome that you got from your dad, which is blue, and the chromosome you got from your mom, which is red, which have the same genes but may have different versions, you've just mixed them up. So that when you hand off one of those or either of those chromosomes to your children, they get a mixture of your parents in that chromosome. This is a really, really important concept, and we're going to work on it until you get it. You may have to spend some time thinking about this. So in Metaphase One, then, we're going to line up the homologous chromosomes, these tetrads that form. So again, if you've got 4 chromatids, right? So let's go to this. You can see 4 chromatids, sister chromatids on each chromosome. There's 4 of them called a tetrad. They're going to line up on the metaphase plate and then pull apart from each other as we move into anaphase 1. This is just like mitosis now, but each chromosome is actually a duplicated chromosome. Each chromosome is a duplicated chromosome that, having gone through crossing over, is actually a mixture of the 2 homologous chromosomes that you started with. Telophase 1 and cytokinesis are going to be the same. You now have half the number of chromosomes at one end of the cell, and it's going to separate, right, and pull apart just like you would do in mitosis. So again, in animal cells, you get a cleavage furrow forming. In plant cells, a cell plate forms, and then you're good to go. So you've got now 2 cells with half the chromosome number. Half the chromosome number, but each of those chromosomes is a duplicated chromosome, and you can see now is a mixture of red and blue. Whereas we started with the chromosomes that are all entirely red or entirely blue. Those cells now are going to proceed directly into meiosis 2. So there's going to be no replication of DNA, nothing else going on. We're going to take that cell and we're going to divide it again. And the key to meiosis 2, again breaking down into prophase 2, metaphase 2, anaphase 2, and telophase 2, that Roman numeral indicating that we're in meiosis and in the second division, is basically just like mitosis. So you're going to see each individual chromosome line up on the metaphase 2 plate, right? And then the chromatids will separate. Chromatids will head to either end of the cell. The cell will divide. In telophase 2, cytokinesis occurs, and now you have 4 daughter cells that are basically gametes with half the chromosome number. But not just half the chromosome number, they're a mixture of the homologous pairs of the original parent cell. Right? So again, in prophase 2, just like mitosis, spindle apparatus forms. Chromosomes are going to attach to that. They're going to move towards the metaphase 2 plate. Metaphase 2 plate, they're going to line up. They're going to attach. Boom. We pull them apart in anaphase 2. Anaphase 2, chromosomes move towards opposite poles. We cut it in half in telophase 2 through cytokinesis. Okay? Now, this is a vast summary of the mechanism of meiosis. We're going to go over this again, basically, and look at kind of the results and why we do what we do in this process.